These are the three key points of principles of biotechnology. We started their discussion in our previous lectures, and today we will move ahead with our discussion. So we already started discussion about identification of DNA with desirable gene. As I mentioned that DNA doesn't have any specific color or shape in the area which is desired by us. So if I am talking about NIF gene, which is responsible for nitrogen fixation, and I want to insert it into E. coli, then obviously there will be no color or N written on that piece of DNA. So identification is required, because then only I can isolate. Next is introduction of identified DNA into host. For that reason, we need to isolate DNA. DNA isolation, as I mentioned, that it is there in your practical isolation of DNA. We discussed roughly about the process of isolation, that what are the ingredients, and then I need to cut it, because if I want to take NIF gene from a nitrogen fixing bacteria, I want the NIF gene only. I don't want the other things, because every cell is having a load of capacity to take load of a gene. Human being can go for replication of genome up to a specific base pair. If I'm stretching my DNA by adding something, then what will happen? We cannot replicate that much long length of DNA. So obviously we have to be very selective while taking the DNA and its insertion. So cutting is very much required. After cutting is done, then there should be insertion. Now the way cutting is difficult, insertion is also difficult. We cannot just take scissor or knife and sit in the table and cut the piece of DNA, because it is not visible, isn't it? So the way DNA is molecular, we will be required scissors of their size. बोलते हैं ना कि जैसा दुश्मन है उसको उसी तलवार से मारो, तो same. So obviously for that we will learn that what is the method. We will use restriction endonuclease for that purpose. Next is after cutting, chalo, it is cut. I got the desired piece of DNA. So next challenge is inserting it into the desired cell. I want to insert it into E. coli. Or in case of treating sickle cell anemia, I want to delete the defective gene and I want to insert the correct gene inside the organism. So for that purpose, I need to during insertion, I need to do what? Take it and push it into some other cell. Can I do it with my hand? So who will do it for me? There are organisms which are called vectors. They are called vectors because they carry. So most of them are actually pathogenic organisms. So they cause disease to us or plants. We nullify their disease causing part and use them as a vector to insert the foreign piece of DNA. So that is the way it is done. And after insertion, obviously, if 1,000 cells are there and I am trying to insert, out of that 1,000 of them will not take my piece of, because no experimentation is complete without standard errors. So how will I identify what are the cells which is actually recombinant and which one no change is there? It's required to identify, no? Because do you think simply insertion of DNA will be enough? No. Its gene expression is also equally important. You know, for gene expression, we studied about the operon concept, that you don't need only the structural gene. You need to have a promoter, you need to have an operator, you need to have a sequence which is repressor. So if I am inserting a gene, I need to insert the gene expression regulation mechanism also there. So I need to not only cut, paste and insert the gene, I need to see it should be expressed, and that too expressed in a desired way. So that can be detected only in the cell in which it is expressed. So there are certain methods by which we can identify, okay, out of this 1,000 cells, only 100 of them are recombinant. Why I am calling them recombinant? Because we teased with their piece of DNA. We inserted something, we deleted something. So they are no more the original one. So we call them recombinant cells. So that identification is required, and once identification is done, next very important thing is the checking that cell is having proper what I can say, transmission of this character, because this is a very costly method, isn't it? So we cannot afford to do it in every generation. We should ensure that once gene is inserted, one recombinant organism is formed, that character should be genetically transmitted to the next generation. If not, there is no point. Understood? So obviously, if not, there is no point means we need to check for the maintenance of that cell line, to study it for several generations. Before I release this in the market, I need to be sure, okay, it is transmissible to the next generation. So we will talk about now the principles of biotechnology. So it is starting with the process of joining and inserting foreign piece of DNA, and that is what is called recombinant DNA technology. Okay. So as I said that playing with the piece of DNA by inserting or deleting, the thing we create is recombinant organism, and the technology is called recombinant DNA technology. So first recombinant DNA that was produced was by Cohen and Boyer in 1972. Okay, and it was inserted in the organism called Salmonella typhimurium. So actually, Salmonella typhimurium had a plasmid, and it is responsible to give it antibiotic resistance. So gene from Salmonella typhimurium was isolated, and it was inserted into E. coli to make it antibiotic resistance, same as the Salmonella typhimurium. Okay, so this is something great that was done, and it was successful up to a very high level. But it's don't take it otherwise. It's not that easy for eukaryotes, because one thing is we are not unicellular, we are multicellular. For example, Gia talked about treating sickle cell anemia. Okay, so sorry, Yesha. So if I want to treat sickle cell anemia, the main problem that I am going to face is in which cell to work. We are not unicellular. Once I am born, I will have millions of RBC. Is it possible for me to remove this defect from each and every RBC? So this gene therapy till date is successful only to certain specific cases, and it will be nice if we can detect about the genetic defect at the initial stage of embryo, when number of cells are less and gene therapy can be progressed. But still it is a dream, but dreams one day comes true, and we can think about it. So now we will talk about the different tools of recombinant DNA technology. So while I was describing the steps of recombinant DNA technology, I told you that we need to cut DNA with the same size of scissors we have. So that is the molecular scissor. It is known as restriction endonuclease. So now, why what are these molecular scissors or restriction endonucleases? So molecular scissors or restriction endonucleases are required to cut the DNA into pieces. So when I am cutting the DNA into pieces, I need to have a scissor that can cut DNA. So what are the characteristics that is required for these things are first important characteristic is it should cut DNA. Second important thing is it should cut DNA at desired place. That means as per my desire. If I am having a stretch of DNA like this, I want only this piece, I want only this piece. You know how the reading frame decides the sequence of protein and how the sequence of amino acids in a protein decides the shape and functionality of the protein. So when I am talking about insertion of a gene, I don't want a mistake of single nucleotide also. Because a single nucleotide mistake can be proved to be detrimental. क्या होगा? रीडिंग फ्रेम शिफ्ट हो जाएगा ना. अगर समझो मुझे टी से स्टार्ट करना है, उससे पहले वाला सीक्वेंस यानी जी ले लिया मैंने, तो होल रीडिंग फ्रेम विल शिफ्ट व्हिच विल बी प्रॉब्लमेटिक. सो हु विल गिव मी दिस? That too free of cost, that too accurately. So what are the source of these restriction endonuclease? The source are bacteria major. Okay. So now the question is, all bacteria are genetic engineers then? Why they are producing restriction? Have you ever thought? Do they go for recombinant DNA technology to produce new stretch of organisms? Then why do they have restriction endonuclease? See, the restriction endonuclease is so why bacteria have restriction endonuclease? That is a question. And the question is for its own protection from virus. Okay, so restriction endonuclease is used by bacteria to cut the foreign piece of DNA inserted in its body. So this is a natural, what I can say, kind of immune system for the bacteria. We should not call it system, it's not that huge, but immune methodology. Because the only organism that can cause disease to bacteria is virus. Others to no chance because they are smaller than bacteria is smaller than them. So this is the way how there are methylation patterns. You know, adenine, guanine, cytosine, thymine can get methylated. What do we mean by the word methylation? Which group is added if I am saying methylation is there? You are going to think and give me this answer. So the formula. CH3O. Dish, what is the formula of methyl group?
Methyl CH3O.
CH3 only, no. Alkyl group. Alkane का एक हाइड्रोजन निकाल दो alkyl. So methyl group is added to adenine guanine cytosine thymine. Now it has a pattern that which number of adenine guanine cytosine thymine will be methylated. So bacteria is having a methylation pattern. Virus, if it is getting inserted into bacteria, that will have a different methylation pattern. So via this methylation pattern, these restriction endonucleases will be able to identify, okay, this is not part of my DNA. This is something else DNA. So I'm going to delete that. And it cleaves that thing at the perfect position. So we are so wise, we are extracting these enzymes, which are called restriction endonuclease, from the bacteria. So now I just want to elaborate the meaning of this word restriction and endonuclease. Okay, so the meaning of the word nuclease means enzyme that can work on why? Because it is working on DNA mainly, but in some cases RNA. Because they are the nucleic acid, that's why it is called nuclease. Nuclease is the enzyme. Now I am coming to word endo. See, two types of nucleases are there. Okay. One type of nuclease is the one which can digest DNA at the end. So if it is able to digest DNA only at the end, I am calling it exonuclease. You know, bacteria is having circular piece of DNA. That's a natural protection of it from the exonuclease. एक्सोन्यूक्लियस उसमें काम ही नहीं कर सकता है. क्यों नहीं कर सकता है? उसके पास फ्री एंड ही नहीं है. ओके, व्हाट इज एंडोन्यूक्लियस? एंडोन्यूक्लियस कैन कट एनीवेयर इन बिटवीन. So it requires free end can cut in between. That's why I am using this word endo. Okay, and restriction, what is the meaning of restriction? Means specific site to cut. So they have a very specific site. What do we mean by site? How they recognize the site? अगर ACCTA है तो वो जहां जहां DNA में ACCTA है वो वही एरिया को कट करे. So it is having that kind of specificity and I am calling it restriction endonuclease. Okay. So they are the molecular scissors with which we can cut the piece of DNA. They can ask you why restriction endonuclease is called molecular scissor. What answer you will give? They cut the DNA molecules and they themselves are very small. So that is about the molecular scissor. I told you about why they are called nucleases, restriction endonucleases. Uh, Till now, 900 approximately restriction endo 900 types of restriction endonucleases are isolated from 230 strains of bacteria. So you can understand how they are used for different sequences. Now, when restriction endonucleases cut these kind of DNA, piece of DNA, they produce two types of ends. Okay, one is staggered end or sticky end. So, what are these? Let us know. For example, when consider there is a piece of DNA. Okay, so in this piece of DNA, so the restriction site is GAT. Okay, so restriction endonuclease will cut here. Now, during cutting, two options are available. One, it can produce a blunt end like this. That means it will cut like A, T, C, T, T, A, T, and then T, A, C, G. This type of ends are known as blunt end. But it's not always like that. Many a times the cutting is staggered and it will produce a sticky end. How? So in this case, one it is cutting here, another case it is cutting here. So it will be having so you can understand that here these press part will be single stranded and here this part will be single stranded. Why? Because it's not cutting horizontally. It is cutting at two different ends. So the way this type of ends are producing single stranded part, we call them sticky ends. Why we are calling them sticky ends? Because they are very well adapted to stick to something else. Understood? So first discovered restriction endonuclease was eco R1. It was discovered from E. coli. That's why it is known as eco R1. So there is a rule of nomenclature of these enzymes. Eco R1 means it is developed from E. coli. I am writing it like that. Actually, it is written like this. Eco R1. So E. coli restriction endonuclease 1. The second discovered restriction endonuclease from E. coli will be known as Eco R2. Okay. So this is the way PAM H1 Okay. So these are different types of restriction endonucleases available. You can see that the first letter will indicate the genus. The second letter, two letters, that is eco, will indicate the species. R is restriction endonuclease, that is strain of restriction endonuclease producing organism, and one is the number one discovered from that specific strain. Okay, so this is how we are talking about the naming of restriction endonucleases. Now, is this part clear? Take your time, have a look, and let me know. So this one is already explained to you, the difference between endo and exonuclease. So I am just moving ahead. The way I told you, there are different types of restriction enzyme. So, while I was explaining the steps of recombinant DNA technology, I told you the first thing is to identify the sequence. Why? Because if you don't know the piece of DNA and its sequence, then you will be clueless in using restriction endonuclease. Why? तुमको कैसे पता चलेगा कि तुम which restriction endonuclease you will use, isn't it? So depending on the sequence of yours in DNA, you will choose the restriction endonuclease. For example, on screen you can see the restriction site of different endonucleases like ALU1, HEI3, that is isolated from Haemophilus influenzae, BAMH1, HIND3, EcoR1. So you can see some of them are giving blunt cuts and some of them are giving staggered cut to produce sticky end. So depending on sequence you want, the gene sequence you want, you have to select the restriction endonuclease. So it is available in the market, and till now whatever restriction endonucleases are discovered, we know the restriction sequence. So that's why we don't use it haphazardly. We use it after knowing. And one more addition I just wish to give here. While we were studying DNA fingerprinting, I taught you about RFLP. That is restriction fragment length polymorphism. So I just wish to describe it in a bit detail here once again. In DNA fingerprinting, if I am taking my DNA, Priyanshi's DNA, and Jia's DNA, and Prachi's DNA, 4 of the DNA we are taking, and maybe this DNA, one male candidate should be there. So out of these 5 piece of DNA we are taking, we will treat them with same restriction endonucleases. Now, the sequence of 5 of our DNA will be different. So if I am using HEE3, that is Haemophilus influenzae restriction endonuclease 3. So in that case, obviously and definitely, my DNA will be cut into maybe 5 pieces. In case of Jia, it may be 10 pieces in Prachi, 2 pieces in Priyanshi, 20 pieces in Dish, 7 pieces, and their length will also vary. So if I am going for agarose gel electrophoresis for DNA, you know, in DNA fingerprinting, I told you about the electrophoresis experiment. The number and location of bands will be different. In Priyanshi, 20 bands will be there. In these 7 bands will be there. In Prachi, how many? 2 only. Jia? 10. My 5. I'm close to you. So number of bands will be different, and depending on their size, position will also be different. So this is a way we can go for DNA fingerprinting also. So we will move ahead. So we will talk about palindromic nucleotide sequences. The best example of palindrome is Madam, I am Adam. I request everyone to write this All caps without any gap. Madam, I am Adam. Write this sentence, everyone. I am Adam. Still, Krishang is using checkered notebook. There is something, मुझे ऐसा लग रहा है कि I should gift you all a New Year's planner, but थोड़ा costly पड़ जाएगा इसलिए छोड़ दे रही हूँ. Now read this sentence forward and backward. You see that it is same. Okay. So always remember such kind of things are known as palindromic nucleotide sequence or palindromes. Understood? So palindromic sequences are very important from the point of view of molecular biology. Why? Because from 5' to 3', whatever the sequence they are reading from 3' to 5', it is same. So in their case, sense strand and antisense strand, there will be not significant differences. Okay. So the question is, if restriction enzymes are giving two types of end, that is sticky end and blunt end, which one do you think is better? Sticky end or blunt end? Okay. So Nakshatra is saying blunt. Why Nakshatra? Sticky. Obviously. Why? Because consider a situation. I just wish to draw here so that you can understand. I have host DNA. And this is my new like DNA I want to insert. Don't you think complementary pair formation will be easier in case of sticky? Did you understand what I draw? No. Okay. Let me write it down, then maybe you will be able to understand. So if this is my host DNA, okay. So it is producing a sticky end. Why? Because this is one strand and this is another strand. So this part is single stranded present over here. Okay. Now I am talking about my second DNA that I want to insert in. So if it is having exactly complementary sequence, so it will be having then how it will be helpful? It will produce a strand like this. Sorry. Understood this concept? So sticky end is always preferable because it is helpful for sticking, because it is at the end some part is single stranded, so that is acting as a tape which will be helpful for letting it allow it to stick with something else. Okay, so um, who is going to join this piece of DNAs? Although they are having sticky end, there will be some gap in between, no? So who is famously used for joining the DNA strand? Who is the ligase? Very good. So DNA ligase is responsible for joining of these DNA bases with sticky end. In screen you can see the diagrams. So this is how, like if the host DNA is having, or the target cell's DNA is having sticky end, and the gene I want to introduce into it is having sticky end, they can be juxtaposed on each other and as a complementary single stranded part, and they can be easily attached with each other. Priyanshi is not happy with the word juxtaposed. I understood from her expression. Juxtapose मतलब वो एक दूसरे के ऊपर फिट हो सकते हैं. Now understood, Priyanshi? Happy? He is not happy. क्या करूं? करो. Is any doubt till now?
No, ma'am.
Great. So that is all about restriction endonuclease or molecular scissors. Now we will talk about the vectors. Okay. Because after isolation of DNA and then cutting of DNA, the next point that is very important is the vectors. May I? So the next very important aim is insertion of. So for that we are using cloning vectors. So why I am using the word cloning here, and what are the meaning of vectors? So vectors are DNA molecules or they can be organisms carrying foreign gene Okay, so they are main role is to carry the foreign gene into something else. So they are just carriers. They are the mediator. Okay. So why I am adding this word like clone? Because they replicate inside target cell to make multiple copies. So that's why I am telling them cloning vector. So bacteriophages can be used as a cloning vector. The plasmids present in bacteria can be used as a cloning vector. Apart from that, yeast artificial chromosomes, phase meat, cause meat. These are the new things. We are not happy with the original vectors. We phase meat are combination of bacteriophage plasmid, sorry, bacteriophage DNA and plasmid. So we call them phasmid. Okay. So various such kind of like things can be used for as a carrier of piece of DNA. So cloning vectors should have some certain characteristics. So that is to be known by all of us. So we will call them ideal characters of vectors. So what are the ideal characteristics of vectors? I request everyone to first listen, then you can write. First, the first character, that is high replicability. Obviously, when I am using a vector, before that I require many of them. Why? Because insertion of one vector into one cell, chances of failure will be too much. So I need to insert at a time many. So that if I am doing it for lakhs, out of that I may get result in 10. It's not an easy thing. Ultimately, we are dependent on cell, we are dependent on vector, we are dependent on DNA, we are dependent on enzyme. And we don't have whip in our hand के चल कर ले, ऐसा नहीं कि भाई MTG के इतने क्वेश्चन सॉल्व करके आना है. तुम लोग क्या सुनते हो कि ये सुनेंगे. Second very important thing is लगा गुड लगने से आगे बढ़ते हैं. Second very important thing is it should have unique restriction site for various restriction endonuclease. यूनिक रेस्ट्रिक्शन साइट, ये जो सेंटेंस मैंने लिखी है, यू शुड बी एबल टू अंडरस्टैंड कि क्या है. सी अगर ऐसा है कि एक वेक्टर है जिसको तुम एज अ कैरियर ऑफ डीएनए यूज कर रहे हो, ऑब्वियसली उस वेक्टर में तुमको कट तो करना ही पड़ेगा, क्योंकि उसके अंदर वेक्टर्स विल बी लाइक अ एनवेलप जिसमें तुम चिट्ठी डाल के ऊपर एड्रेस लिख के भेजने वाले. बेसिकली दे आर लाइक दैट. सो ऑब्वियसली वो चिट्ठी का जो एनवेलप है वो अगर चारों तरफ से खुलने लगे तो क्या होने वाला है, खिचड़ी. तो 4 लोग अपने चिट्ठी डाल के भेजेंगे, जिसको मिलेगा उसकी हालत सोचो. ठीक है. सो ऑब्वियसली यूनिक रिस्ट्रिक्शन साइट मींस अगर इको 1 का रिस्ट्रिक्शन एंडोन्यूक्लियस का साइट हुआ तो इट शुड बी मैक्सिमम टू 1 or 2 प्लेसेस एट दैट वेक्टर, ताकि मैं जब कट कर रही हूँ ऐसा ना हो कि मुझे कट इधर करना है कट उधर हो गए. दैट शुड नॉट बी देयर, नो. बट ऐसा भी नहीं है कि एक ही रेस्ट्रिक्शन एंडोन्यूक्लियस के लिए हो. फिर क्या होगा, मुझे इतनी पीस कट करनी है, इतनी पीस की ही ऑप्शन मेरे पास रहेगी, दूसरी ऑप्शन नहीं रहेगी. सो रेस्ट्रिक्शन साइट ऑफ एंडोन्यूक्लियस इज पर्टिकुलर. जैसे अगर एक स्पेसिफिक एंडोन्यूक्लियस GATA में कट कर रहा है तो वो GATA में ही कट करेगा. तो अब GATA इस वेक्टर में एक या दो जगह पे ही होनी चाहिए ताकि कटिंग के वक्त कंफ्यूजन ना हो. दूसरा, अदर देन GATA दूसरे इंट्रोन्यूक्लियस के लिए भी सीक्वेंस होनी चाहिए ताकि मैं मेरी सुविधा के अनुसार इनको कट कर सकूं. ठीक है. थर्ड थिंग इज इट शुड बी हैविंग हाई ट्रांसमिसिबिलिटी. मतलब अगर मैं इसको इसके अंदर जीन डाल के इसको टारगेट सेल के पास भेज रही हूँ, so it should have ability to transmit this gene to next. इसीलिए जनरली वेक्टर्स के तौर पे उनको लिया जाता है जो डिजीज कॉजिंग है, बिकॉज दे आर एक्सपर्ट इन गोइंग इनसाइड द सेल. ठीक है. फोर्थ थिंग इज इजी टू कल्चर. So ideal vector should have, all of them should have this type of characteristics which should be present in them. Okay. So cloning vector are the methods by which we can transmit the desired piece of DNA. If you want to take a screenshot, you can. So we will look into different types of cloning vector. First example is plasmid. You know, bacteria have extra chromosomal genetic material which is circular small piece of DNA outside their nucleoid region, which is called plasmids. And if you must have studied in grade 11 that there are cells called F plus and F minus. The F minus cell, that is fertility minus. You studied about conjugation. These studied about conjugation in grade 11.
नाम तो सुना था, पता नहीं अब.
So plasmids are very much important for bacteria. They are responsible for giving them extra characteristics like antibiotic resistance. They are responsible for synthesis of substances like nopalin, octapine, opine. These are extra chemicals synthesized by plasmid. So the cell which has plasmid is known as F plus or fertility plus, and the cells which doesn't have plasmid is known as F minus. If you could have asked grade 11 students, I taught them there is a process which is called bacterial sexual reproduction. Although we cannot call it reproduction in true sense because there is no increase in number of organisms, but there is a method called conjugation by which the F plus cell will make a copy of its plasmid and transfer it to F minus cell. This process is called conjugation. तो अब F minus मतलब उसके पास प्लाज्मिड था नहीं, उसको F plus ने एक कॉपी दे दी, तो अब वो क्या हो गया, खुद भी F plus हो गया. So this plasmid, which is known as extra chromosomal genetic material, I said it is a small circular piece of DNA, can be used as a vector. ठीक है. So PBR322 is the plasmid isolated from E. coli and it was used as a vector. Okay, so you must have seen that E. coli is very nice. So many things it gives to us. Okay, so plasmids that we choose for recombinant DNA technology should have high copy number. Means they should be able to produce more copies because obviously otherwise they will not be able to multiply. Okay. Now, other than that, bacteriophages, obviously they are best source because they are naturally infecting us. So there are virus, animal virus like the adenovirus or there may be plant virus like tobacco mosaic virus. They can be used as a source of vector. Okay, so when the vectors are multiplying in the host, they will make multiple copies of the genes also, isn't it? Consider you have one bacteriophage, you inserted your gene into it. When bacteriophage will make 1,000 copy of it in the cell, it will make 1,000 copies of the inserted gene also, no, in the cell, isn't it? So what are the features of cloning vectors and others that we are going to see in our next lecture, where we will discuss this in detail and move about this chapter further. Okay, so I wish to say bye today. We will meet in our next lecture and discuss with the vector further.